Welcome everyone who has joined. This is, if you're joining for the first time, we at Claret have a webinar series called as the Serious Talk, where we showcase work by the users of Claret Bio products and also some internal presentation where folks at Claret showcase new products, new technology, pipelines, and so on. And keeping that in mind, today's talk is a slightly different talk because we've never done a bioinformatics focused one before. Here we'll have our star bioinformatician, TobinGroth. He will give you an overview of some of our pipelines. These are data analysis pipelines that we offer for free on our website. These are all open source software. And in this talk, Tobin is going to go over installation, running, usage, etc. I'd like to invite Tobin again. Tobin is a valuable member of the Claret Bio team. He got his master's in bioinformatics from University of California, San Diego, and has been central in overseeing a lot of the data analysis that we do internally, and also in development of the pipelines that are available for general use. With that, I now punt this to Tobin. Thank you, Tobin, for agreeing to give this talk.
Yeah, I'm very happy to be giving this talk. And yeah, as Varsha's mentioned, my name is Tobin. I'm a computational biologist here at Claret, and I actually have helped build this software as well as some of the other softwares that are available from Claret Bio. And in this talk today, we're going to mainly be focusing on a general overview of Seriously Run. So as Varsha mentioned, we'll be talking about installation, how to run, and then ultimately what the pipeline will be outputting, which will be what be in your hands after you run this pipeline. And so there are multiple offerings from Claret Bio. We offer three different types of software, Seriously Run, Seriously UMI, and Really Run. Each of these are slightly different. We'll be focusing exclusively today on Seriously Run, pardon me, which focuses on using DNA samples that have been run through Seriously Library Prep that take in FASTQs files as input, and you're able to run the software on samples that also contain UMI sequences. Alongside that, we also offer Seriously UMI, which is a software mainly focused on when you're getting BCL files right off the sequencer and need to demultiplex those UMI containing sequences to get to those FASTQ files. And then lastly, our most recent software offering is Really Run, which is the RNA equivalent to Seriously Run and is mainly used for running FASTQ files that were generated from RNA input and using the Really library prep. And so today, as I mentioned, we'll be mainly focusing on Seriously Run, but we do plan on offering other webinars in the future regarding these other products. And so one of the things we want to talk about today is just to highlight where exactly Seriously Run will fit into the analyses that you're performing. And so within the general workflow of your analyses, you start with your input DNA. Obviously, you're going to be using Seriously Library Prep, best library prep around. And with this, you're generating your DNA library. Ultimately, you sequence that to generate your paired and FASTQ files. And then once you have these FASTQ files, this is where Seriously Run can be used to generate, basically going from FASTQ files right to stacked analyses files. Right? So this is basically a tool that makes it so you don't need to worry about finding different software tools to use or writing data pipelines to generate these files. Seriously Run offers all that in one package. And so this is providing an overview of what's going on behind the scenes in the Seriously Run software. And so I've highlighted sort of the main components of the pipeline as well as brief diagrams of what's really going on with these tools. And so the first thing that has to happen once you're working with raw FASTQ files is adapter trimming. And so adapter trimming is really important because sometimes, as you can see in this diagram on the top left, you have your insert sequence, which when it comes off the sequencer has adapters flanking it on both sides. And so once the reads are generated that start at one end of the insert sequence, sometimes if the insert sequence is shorter than the amount of cycles that are being used by the sequencer, you can have some read through into the adapter sequence. And so if you try to map these sequences or align these sequences while they contained the adapter sequences, you'd have a lot of trouble and your mapping scores would be quite poor. So because of that, we do adapter trimming, which searches for the portions of the adapter sequence in the reads and removes them. And this allows, for downstream use, you're able to map much better. And in this pipeline, we use cutadapt. Cutadapt, there are many tools that are offered, but cutadapt is a great tool that runs very quickly and it works great with Illumina sequencing results and is a good tool that can hold on to the discarded reads and trims the adapters from the discarded reads and helps with dimer interpretation as well. And that's one of the reasons why we employ it in this pipeline. Following adapter trimming, you're going to be performing alignment, where, as you can see in this diagram on the left, on the right, you have your set of reads and your reference genome, and the alignment is where we're finding the best location for those reads to map to on the reference genome. And so this is really this is what is like a crucial part of most pipelines, where you have all this data, but you need to know where it's coming from exactly on the reference genome. And so we employ BWA-MEM in our pipeline. There are many different alignment tools, but BWA is one that we've been using for quite a while. And BWA-MEM is a local aligner, meaning that it can perform soft clipping. I won't go too much into details here, but BWA-MEM generally has much higher mapping rates than other tools, such as BWA-Align or Bowtie. And so in our case, because we're just interested in aligning and having as high of alignment rate as possible, and we're okay with some soft clipping, we use BWA-MEM for this reason. Once you have your reads aligned to the reference genome, it's important to perform duplicate marking. And duplicate marking is important because you can sometimes have reads that are the exact same that have, because of sequencing issues, are exactly the same on the exact same read, mapping to the exact same position, and there's not actually going to be, you want to consider those reads to be unique reads necessarily because they are exact duplicates. And so we use Picard duplicate marking because it does not remove the duplicates from, or you have the ability to not remove the duplicates from the BAM file because we don't necessarily want to get rid of them, but we do want to be able to exclude them from analysis, from downstream analyses, since we only want to be looking, excuse me, at unique reads. And so once we've performed all three of these steps, adapter trimming, alignment, and duplicate marking, lastly, we will use custom stat scripts and SAMTools flag stats to generate general stats regarding the BAM files and the rest of the output files that are done or that are generated from this pipeline. And one thing I should also mention as well is that during duplicate marking, Picard also outputs a stats file as well. So you get a plethora of stats files that are at your disposal. And so there's going to be sort of three main components to this talk. We're going to go through installation, where we'll be going over the necessary dependencies for srslyrun, as well as how to install srslyrun itself. And then the next step, we'll be looking at what we need to run srslyrun, and then how we are going to be running it. And then lastly, just some steps to show you all exactly what to expect from the pipeline and how to make sure that it's run properly and that you are getting all the files that you expect. So firstly, we'll be starting with installation, as hopefully installation should be a very boring process for any piece of software. As soon as installation is exciting, you're looking to have a rough time. And so the dependencies that are needed for srslyrun are going to be Python, which is a open source programming language that is employed in many, many different labs and companies and is really useful for it has access to lots of different types of libraries and is the backbone for how we've coded srslyrun. And the next other dependency that you would need is Mamba, which is a virtual environment packaging software. And so we use this to make sure that you don't need to install all the different tools needed to run the pipeline. Mamba will do that for you. You can also use Conda, which is an older version of Mamba that does not. So Mamba uses C. I won't go into the details of this right now, but Mamba is faster. And if you use Conda, it will run perfectly fine, but it will just be much slower. So all you really need to do is install these two dependencies. And then you should be able to run pip install srslyrun. And that, in a perfect world, that will work. But, not every computer is, set up in a perfect way, and there are always issues, technical difficulties. Believe me, we've dealt with these many times. And so I also want to offer another solution if this straightforward pip install srslyrun does not work. So this is where something called virtual environments can be super helpful. And so typically installations go wrong when you have conflicting package versions or you have software that is older and is causing issues, it hasn't been up to date. These kinds of things can always happen. But fortunately, there's a super helpful tool called Python VM, which creates a virtual environment that is totally separate from all of the other Python packages in your system. And so this makes it so, for example, if you were to create a Python virtual environment, install srslyrun into it, it would be fully separate in its safe little bubble away from all the other Python packages that could be causing these conflicting issues where installation was having trouble before. But what's great about this too is that even though you have your package separated from all of the Python packages, it still has access to all the non-Python packages present on your computer. So as you can see in this diagram here, if we were to create a Python virtual environment with srslyrun, it would still have access to Mamba, as would other software. And so this is a great way to sequester particular packages in their own little bubble, just so that they can always be ready to go when you need them. And you can still install stuff willy-nilly and not worry about how it affects that specific virtual environment. And so I'll go over the steps that are needed to create this virtual environment with srslyrun in it. So the first thing you'll need to do is simply create this virtual environment using this python-m vn command. And then after that, you'll just have to pass the path to where you want to have this virtual environment created. And so this is usually, you can put this wherever you want. If it's only going to be you using this, you can put this in your home directory if you have some sort of like sandbox that you use. But if you want this to be accessible to multiple users, you should make sure that you have the path to this virtual environment in a place that's accessible to other people as well. So you only need to use this one command to create it. And then once you've created it, you need to activate it by using the command, the source, and then the path to that virtual environment, and then that bin and activate part of the path as well. And those files will be automatically made simply by creating the virtual environment itself. And so once you've activated the virtual environment, you've now stepped into that virtual environment and you are now in this new bubble of Python where you actually do not have any packages installed. It's like a very fresh installation of Python where you're safe from the evil conflicting packages that may have caused you problems in the past. And so a good thing to do just to make sure that this virtual environment was created successfully is by checking that pip is installed or is upgradable. And so this is just a kind of good sanity check to make sure that everything's working as intended. And if this doesn't work or has issues, it's super easy. You just kind of go back to square one, delete what you've just made, remake it again in the same steps, and hopefully this will work better. Sometimes, things can go a little wrong here and there, but if this step does not work, it's super easy to just recreate and kind of go step through these again and then try again. And if that does work, then you're ready to install srslyrun in a similar way that we had outlined before, but this time you're now doing it in the virtual environment and you've added this python-m pip install srslyrun. And so what's really important here is to make sure that you use this command from within the virtual environment so you've properly activated the virtual environment itself. And so once you've installed srslyrun into this virtual environment, you should be able to use srslyrun without any issues. And what's important to note too is that once you are done working within this virtual environment, it's always a good idea to deactivate it using this deactivate command, and that puts you back out of the virtual environment into your general computer space where your previous packages and everything else was installed. And so it should be noted as well that anytime that you need to run srslyrun, you will need to activate it, do your srslyrun command, and then deactivate it once you're complete. And so this can be a bit of a tedious method sometimes. It can seem a little daunting to sort of do this, but in actuality, it's probably best practices for installing software like this so that it can again live in its own bubble where it's not being affected by the other packages that you're installing onto your machine. And so now that we've installed srslyrun properly, the next thing we want to do is kind of talk about how to run it and the results that are being run. And so what's actually needed for srslyrun? You don't need a whole lot. As I mentioned before, all you need are those paired end FASTQ files, which should be coming off your sequencing run. And then the other thing that you need is the reference FASTA file that corresponds to the species, or like the input DNA that was used to generate those FASTQ files. And so these can almost always be found online, either at NCBI's RefSeq, Ensemble, or the UCSC genome browser. So these are super useful resources to find whatever species you're looking for. More often than not, they'd be found there. You'd be hard pressed, I'd say, to find one that's not there, and at least in one of those resources. But these are the only things that you really need. The files that you need to run srslyrun are these FASTQ files and this reference FASTA as well. And so once you've collected these files, the next step is, running our command. And so I'll go over, what needs to get run, or how you are going to be running this, as well as the different options that you're going to be needing to use. So with srslyrun, we have two types of options. We have required options and then optional ones. Required ones are going to be needed every single time you run the srslyrun software. And so what we'll need to do first for every single run, you'll start with srslyrun samples. You can follow that with the required options, which is this dash dash reference, which is simply the path to your reference file. And this file, again, must end in .fa or .fasta. And usually I would recommend too to do the absolute path so that you're starting from whatever the highest directory is in your system and passing the path all the way to that reference file. And that can just avoid any issues with some sort of path interpretation by the software itself. And the next thing you're going to need to use is to specify which libraries you want to be run through srslyrun. And so that can either be used with dash dash libraries, which is just a comma-separated list of the library IDs to be analyzed, or you can use libfile, which will contain those exact same library IDs, except this time it's on one ID per line in the file, and you just have to pass the path to that file that contains those library IDs. We also have optional flags as well, such as dash dash fastqdir, which is the path to the directory where the FASTQ files are contained. This defaults to the directory that you're currently running the software from. So if you're in the directory that contains the FASTQ files and you're running srslyrun from there, you don't need to specify this. And the same can be said with results.dir, where you have the path to a directory where the results want to be, where you want the results to be. And again, this defaults to the current directory. So if you want your results and your FASTQ files to all be in one location, you don't need to specify either of these as long as you run srslyrun from that location. And then lastly, if there are UMIs present in your sequencing data, then you can use this dash dash UMI flag. And this will run some additional tools. I won't go into too much detail about this option today, but is a good tool to be used, following if you've used seriously UMI to prepare your BCL files and demultiplex them properly. This is what you'd want to be using following that as you're using srslyrun for the next steps. And so here's an example of how you can be running srslyrun. And so this is assuming that I'm running my command from this directory where the FASTQ files are stored, this test FASTQ directory. So this is where I'm going to be running srslyrun from. And so I'll use srslyrun samples, and then I'll pass the libraries that are being run. And so in this case, we can see that the name kit1 is the prefix for my FASTQ files. And so we'll use that as the library ID for these FASTQs. And it's important to note too that the libraries should always contain a prefix that's present in your FASTQ files, or else the software will not be able to connect the library name to the FASTQ file itself. So it's always important to note that all your FASTQs, if you have multiple of them, multiple libraries to be running through, that they have some amount of unique identification at the beginning of them so you can properly distinguish them with different library names. Secondly, we'll have the other required option, which is the reference flag. And that is, again, the path to the reference that you want to be using. In our case, this is the E. coli. And then lastly, you'll have the results.dir. In this case, you don't need this because you can simply have the output be the current directory that you're running from. But in our case, I wanted the results to go elsewhere, so I passed this path of where I want the results to be. And so, for example, if we weren't running this command from this test fast queue, we would then need to add what you can see on the bottom here, this fast queue dir and then a path to that test fast queue directory. And so this is an example of how you can run srslyrun. And then next we'll be going over the files generated by srslyrun and sort of what you can look like in your example results. And so it's important to see here is, as I mentioned before, let's go back to this really briefly. The resultster is this non-UMI results, libraries is kit1, and then the ultimate structure for the output of this data will be all of the library results beneath that results directory, which as you remember, it was non-UMI results. And then for every library, there will be a directory with the library ID that contains all of the results beneath that that are all prefixed by the library name. And so as you can see, there are a number of results files that are not just files themselves, but also the logs associated with different tools. And then there are also workflow files, which are not too important, but it's always good to make sure those are there, and they should always be within the results directory. And so this is a good step too to double check that everything. And so this is, sorry, this can be used as a good resource for when you run this yourself and you want to make sure that everything ran correctly. Firstly, you can double check that all of these files are present. So for every single library that's run through, there should be all of these files present, as well as all of the workflow files present that should be present in every results dir. And then the other thing that you can check too is looking at the different log files. And so log files are incredibly helpful to understand how tools were run or if there's any issues when running particular tools. The best log file, most of these log files, if run correctly, should be empty. And if there are issues, there will be information in there regarding the tool itself. And so this is always, looking through the log files is always a good way to double check whether or not the software was run correctly. And so next we'll be talking about some of the different stats files. And so from I'll just go back here one very briefly. We have the kit1.stats file. And so that's sort of the main results that we are generated from like a custom Python script that takes sort of in all of the data that's generated and tries to create a nice summary of the data itself. And so this takes in the pairs too short, pairs kept from cutadapt, as well as all the mapping information from BWA mem and then the samtools flag stat command. And then lastly, we'll have all of these Picard stats that are coming from Picard's duplicate marking software. And so hopefully your stats will look a little bit better than mine. Ideally, you'd be having, a much higher percentage of your BAM file being mapped. And generally, when you see this is just sort of a small detail, but when you see this Picard estimate library size as -1, usually that means that you did not have enough mapping to generate an actual library size estimate. And so that can always be an indicator that something's gone awry in your analysis. And so in this case, we just used some random test data on using that E. coli FASTA file. And so this is sort of why you're seeing such funky data. But on the right side too, you can also see the figure generated using, this is the molecular length plot, which highlights all of the lengths of the data present and the amount that have mapped versus unmapped. And so in this case, we see a very high mapping. You can kind of just see this is generated from E. coli data. You can see that it's genomic E. coli data. So it's sort of just this big, nice curve. And you can see the amount of the library at each length. And so and this can be helpful too if you're looking for if you expect to see something at a certain length and you're seeing in this result that it's not quite the same, that's always a good way to understand, how successfully your library was run through the sample, and just to give yourself a good idea about, how well you ran it itself, and if there could be adjustments like, okay, maybe this wasn't the right reference file to use because things aren't looking as I expect, and sort of adjusting and so then you can always rerun based on how you've recognized the results to be. And so that basically covers all you really need to know regarding how to run srslyrun and the files that are generated, as well as how to double check to make sure that everything was run properly. And so there are some things that we want to be doing in the future for Claret Bio in regards to the software. The first thing that we plan on doing is modifying our srslyrun, run, and srslUMI packages so that they're available not just through pip, but through also Conda or Mamba, because this offers a little bit more flexibility in what's installed and also is generally just an easier process for installation. The other thing that we have on the in the future is going to be video tutorials for the installation of these different tools. So this will be a more focused approach about, a screen share showing exactly what commands to run. And it'll be very similar to what you've seen today, but with, a terminal screen and, one of some probably me typing out the different commands and showing exactly what to be expect and, the exact process for installation of these different tools. And then lastly, we also plan on having another informatics webinar, which regarding and more focused to Seriously UMI, which was again the tool that was mainly used immediately following sequencing. So before FASTQ files have even been generated, you need to demultiplex that. And when you have UMI sequences present in your reads, this can be a bit of a tricky task. And so Seriously UMI is the tool that can convert those BCL files in that sample sheet into usable FASTQs that are aware of the UMI sequence and add it as a tag to that FASTQ file. And so with that, that kind of covers everything that we hope to talk about, but we're still planning on doing the Q&A session as well. And obviously, if you do run into any troubles in the future regarding running Seriously Run or any of the other softwares, please reach out to our technical support team. Generally, the software issues fall onto my plate. So if you do have any questions, you'll most likely be hearing from me, and I'm always happy to help our customers